PhD in Microbiology and Immunology from Indiana University, where she worked on the role of CD1D1 and NKT cells in antiviral immunity. She then continued her training in immunology, still at Indiana University, as a postdoc before moving to Johns Hopkins for her second postdoc in cancer immunology. Dr. Webb then became a faculty member at the University of Maryland School of Medicine, where she is now an associate professor with tenure in the Department of Microbiology and Immunology. She is a leading immunologist who is recognized for her studies focused on the modulation of NKT cell activation for cancer immunotherapy. And her lab currently explores the mechanisms that NKT cells utilize to recognize and destroy tumors, how cancer suppress these anti-tumor NKTs, and novel therapeutics to restore NKT cell function. Dr. Webb is not just a great scientist, she also has an entrepreneurial spirit about her. She has thus far generated 4 patents and is the founder of Webb Cures, a biotech company that focuses on NKT cell-based therapies for cancer. And what else is apparent from her CV is that she is a passionate and committed mentor. She has mentored dozens of trainees at various stages of their careers, ranging from high school to junior faculty. She has also been an active leader and participant in the University of Maryland STAR Prep, a program designed to mentor and guide post-bachelorette underrepresented minorities to enter the competitive PhD programs. And she also is co-leading a T32 on cancer biology. And with that, I just want to put out a reminder that all participants that want CME credit to refer to Dr. Kota Gomez's message in chat to gain that. And thank you, Dr. Webb, for taking the time to meet with us. And we're all really excited to see what you have to share. The floor is yours.
Oh, thanks so much. Thanks, Joey. That was an amazing introduction. Hopefully I can hold to it. And thanks so much, Eduardo and Adela, for the kind invitation. I'm so happy to share with you some of the work that we have going. See if I can pull it up really quick. Going to adjust the pointer, and you can hear me okay, right?
Yes, I can hear you.
Okay, and thanks, Mercedes, for joining. Oh, I'm so happy to see you.
Yeah, I'm so glad to see you.
You as well. And I'm going to tell 3 short stories. In the last one, you can see how we were able to turn around the situation. So the last time we talked, I told you about the challenges, but hopefully now you'll see that we were able to really turn things around.
I'm really, really happy to see you here today.
Oh, thanks. Thanks so much. Okay, and so my talk is really geared for the students. So if you have any questions, please feel free to let me know. I'm going to tell 3 short stories. I was hoping to pick little vignettes or little snippets that you would be able to take something from. So overall, my talk is focused on NKT cell development and activation, because we want to modulate NKT cells for cancer immunotherapy. So for those of you like pictures, this is my white blood cell killing cancer, and that's what we want, right? We want to induce potent anti-tumor immune responses. So I have to disclose that I work with several companies, but I won't go into any of that work today. The only reason I'm showing you this, well, I need to show you, but to say, work hard, you never know where life will take you. I never imagined I would do things like that, but it's happened. So the focus of my talk is on cancer immunotherapy, and that's because it's been heralded as the breakthrough of the year numerous times. So these are old covers of scientific journals, and someone else has called it the greatest thing since sliced bread. But what I want you to focus on is the issue of time, where it says, what if your immune system could be taught to kill cancer? And again, that's our goal, right? We want to induce potent anti-tumor immune responses. So it says inside the brutally selective, and that's because, as I'm sure you're aware, cancer immunotherapy only works in 30 to 40% of patients. And it can be hugely expensive, meaning it can cost $30,000 to $50,000 per cycle, per treatment. And you could require 5 cycles. Or if you look at CAR T-cell based therapy, your take-home bill can be upwards of $1 million. So that's really expensive. But what we want to focus on are the life-saving trials of immunotherapy. So the work in my lab is focused on developing therapeutics that could be broadly applicative, all right, and maybe not as expensive, but again, we want to save lives. So what are NKT cells? So natural killer T cells, they have characteristics that are of both the innate and adaptive immune response. So NKT cells are T cells. They do have a T cell receptor. And if you look here at classic CD4 and CD8 T cells, which recognize peptide antigen presented in the context of MHC molecules, antigens that are recognized by the T cell receptor on NKT cells are lipids. So CD1D is the antigen presenting molecule that's structurally similar to MHC class 1. It traffics like class 2. But again, it presents these lipids to NKT cells. And remember I said I wanted to make a therapy that's broadly applicable. So everyone in here has NKT cells. And they can also be called iNKT cells or invariant NKT cells, the ones that we work on, type 1, because they express a semi-invariant T cell receptor. So in mice, it's V alpha 14 that pairs with a limited number of V beta chains, and in humans, it's V alpha 24. So again, if we can target this key population of immune cells, we can use it across the board. At least that's what we hope. So hopefully you shared a little bit of my enthusiasm for NKT cells, and they are very promising for cancer immunotherapy because not only do they directly kill tumor cells, they make a plethora of cytokines. They quickly, after activation, produce high levels of interferon gamma and other cytokines that can induce the maturation of dendritic cells, as well as help CTLs or cytotoxic T lymphocytes, as well as NK cells mediate their effector functions. So because these cells, again, can directly kill tumor cells, as well as enhance the effector functions of other immune cells, they're thought to be really promising, but this is slightly taken away from because they are suppressed in many cancer patients. So if you look across the board at cancer patients, it seems to be independent of tumor type or tumor load, NKT cells are reduced. And so that's why this has limited their efficacy or their ability to be used in clinical trials. So what we want to do, again, is to understand why are NKT cells reduced in cancer patients, and then develop strategies to restore their function in order to move the field forward. So the first story that I'm going to tell you about today is focused on ovarian cancer. And that's because it's the most lethal gynecological malignancy in the US as well as worldwide. And that's because while more than 70% of patients, or the majority of patients, initially respond to treatment, the majority develop this recurrent chemotherapy-resistant disease. And that's because the symptoms of ovarian cancer are really mild. You have nausea, bloating, back pain, and most people experience nausea, bloating, back pain, right? So you wouldn't necessarily think that it's ovarian cancer. So this leads to patients being diagnosed very late. And so this leads to the 5-year survival rate of stage 3 patients being less than 25%, and for stage 4, only 5 to 10%. So this is a really, really poor outcome. But what we do know is that ovarian cancer patients often present with ascites, and that's fluid that's collected in their abdominal cavity. So when I was a postdoc, we had this really simple hypothesis. We wondered if the cancer secreted inhibitory factors in this fluid, in this ascites. And so that's what we saw to test. So again, we collected ascites from a panel of ovarian cancer patients, treated our CD1D expressing antigen presenting cells with this ascites, and then looked to see if NKT cells could be activated. On the x-axis, I'm showing you 3 different ovarian cancer patients, and then our simulator cells that express CD1D were treated with media. Here's a negative control. On the y-axis, I'm showing you percent inhibition. We were looking at NKT cell inhibition by measuring IL-2. So of course, when we treat with media, there's no inhibition, and each bar is a different NKT cell line. And so what I think you can appreciate is that if you look here at the black bar and compare that to like the gray bar, we saw a 50 to 100% inhibition and the ability of our antigen presenting cells to stimulate or activate NKT cells after being treated with ascites. So this was just 4 hours. So if you take your antigen presenting cells, your stimulator cells, treat them with ascites, within 4 hours, they lose the ability to activate NKT cells. So our hypothesis was correct, right? There was something there that was inhibitory. So we wondered what it was. So we took a moderately inhibitory ascites, you see it's moderately inhibitory, right? And extracted the lipid fraction because NKT cells are activated by lipids. So we thought they could also be suppressed by lipids. And again, we're looking at percent inhibition here. And you see that most of the activity was in the lipid fraction. So then to further identify what it was, we ran TLC, old school thin layer chromatography. So this is our gangliosides, our standards, because we isolated the polar lipid fraction and looked at the phospholipids and the glycolipids, and the majority of the inhibitory effect was in this ganglioside lipid fraction. And we found that as the samples had increasing amounts of these gangliosides, we saw a higher level of inhibition. So this was exciting, right? We found that the patient ascites contained an inhibitory lipid species that was blocking the ability of NKT cells to be activated. So then we bought those lipids from Maitreya, and for time, I'm just showing you two here. So this is the ganglioside GD3, and we saw a nice dose-dependent inhibition of NKT cell activation when we treated our antigen-presenting cells with GD3. This is in contrast to GD2, which is just missing one sugar. There was no effect. So this strongly suggested that the inhibitory factor was GD3. And so all of this had been done in vitro, so we wondered in vivo what would happen. So we took C57 black 6 mice, treated with alpha-gal-ser, a potent NKT cell agonist, and you see we get nice NKT cell activation. However, when we added increasing concentrations of GD3, we lost NKT cell responses in vivo. And then you may be wondering, is this specific for NKT cells? So actually, we also performed ICS, intracellular cytokine staining, and we did find that this reduction in cytokine production was due to a loss of NKT cell responsiveness. And for those of you who like structures, here's the structure of GD3. So this is exciting. This is when I was finishing my postdoc and just starting my lab here. And so it was really nice to find this inhibitory factor. And then I had a graduate student who thought, okay, you found one factor, but is that it? So she looked at VEGF, or vascular endothelial growth factor. And that's because VEGF levels in ovarian cancer patient ascites are much higher than that found in other solid tumors. In addition, high levels of VEGF inversely correlate with survival in ovarian cancer patients, but it does directly correlate with invasion and metastasis of these cells. So that's from the cancer side. But if you look at the immune system side, it's known that VEGF causes the recruitment of regulatory T cells, or Tregs, in MDSCs. So VEGF causes the recruitment of these suppressor cells, has been associated with decreasing CTL function, as well as enhancing disease and helping the tumor to develop a blood supply. So we looked at it. And so this is a new patient, panel of ovarian cancer patients from our cancer center here. So we first looked to see if VEGF was present, and it was in the ascites. This is non-malignant ascites. It wasn't there. This is recombinant protein as a positive control. So VEGF is present in the patient samples. We looked to see if it was secreted by ovarian cancer cell lines, and it was. Then we bought recombinant protein, treated our antigen presenting cells with VEGF, and found that it blocked their ability to activate NKT cells. So VEGF is present and it's suppressive. So can we block it? So we used two different methods. We used Avastin, an antibody that binds soluble VEGF, and we treated human ovarian cancer cell lines with Avastin or Genenstein, a tyrosine kinase inhibitor. in order to block the production of VEGF and the culture supernatants. We took those culture supernatants, treated our antigen presenting cells, and in both cases with Avastin and with Genenstein, when you block VEGF by increasing the concentration of both drugs, you restore the ability of NKT cells to become activated. So then we wondered if there was a link between GD3 and VEGF. So the top is just showing you our controls. So if you look at the red open histograms from both ovarian cancer cell lines, you see that we can detect GD3 by staining. And then below, if you compare the vehicle control to Genenstein, or the blue open histograms to the red, you see a leftward shift. So if we block VEGF by treating our ovarian cancer cells with Genenstein, we get a decrease in GD3. So this was exciting. This was the first time in ovarian cancer or in a human malignancy, we found a direct link between an angiogenic factor, VEGF, and an immunosuppressive lipid. Oh, my slides slip a little bit. So that was just a quick story just to show you how you can take a basic question, wondering about immunosuppressive factors being secreted by a tumor. So that's what we found. We found that ovarian cancers shed both VEGF and GD3, and this blocks the ability of antigen presenting cells to stimulate or activate NKT cells. And we've also looked directly at NKT cells and found that VEGF directly inhibits their function. So what are we doing from there? So I had a STAR prep student, post-bac, who developed ovarian cancer 3D models, because we found that these factors also induce the differentiation of monocytes to MDSCs. And so we wanted to have a 3D model to look at these MDSCs in the tumor microenvironment. And then I've had 2 high school students recently work on diagnostics. So no matter what your stage, you can have a significant impact on science. And then we also have an interest in cancer health disparities. And we were looking at SNPs, SNPs, single nucleotide polymorphisms, and in immune-related genes. So in checkpoint, like CTLA4, we looked at CD28 signaling, in African Americans versus European Americans. And we were looking for a functional result. And so we decided to think bigger. And we worked with Dr. Jones, Lisa Jones, in the School of Pharmacy. And she's developed a method called FPOP, or fast photochemical oxidation of proteins, in which you can treat cells with a drug, label those cells. And when you label them, you're labeling the proteins within the cells, so you can identify post-translational modifications by mass spec. And so what we've done is, again, you have your proteins within the cell, and we can do in-cell, live cell labeling of the proteins, then digest those proteins and run on mass spec. And so we asked the question of what happens if you use a commonly used drug in cancer like Gleevec in different populations. And what we found were the changes in the proteins were completely different, or significantly different, when you compare cells from European descent versus those of African American descent. So this is very striking. Can you believe this? A commonly used drug that's FDA approved differentially modifies proteins based on ethnicity. So this makes a strong call for personalized medicine. And so again, you can ask a very basic question: do drugs work the same? And find out, surprisingly, that there are striking differences in things like pyruvate kinase, which you wouldn't necessarily imagine. So we've done this in triple negative breast cancer, as well as in ovarian cancer cell lines and primary cells. So stay tuned for our updates on that. Okay, so I've talked to you about our ovarian cancer story. And showed you that there are obstacles for anti-tumor immunity, namely VEGF. Again, we found that ovarian cancers secrete inhibitory factors such as GD3 and VEGF, which blocks NKT cell responses. And there are other mechanisms in play, right? You can have a lack of co-stimulation, you can have tolerogenic cell death, the tumor microenvironment can be extremely hypoxic, you may not get activation of cells, right? Because you could have reduced MHC expression or inhibitory molecule expression after activation of T cells like CTLA-4 and PD-1. And so we wanted to probe this to see what mechanisms could be impacting NKT cell responses. So again, as I mentioned in the introduction, NKT cells have characteristics that are of both the innate and adaptive immune response. So from the mouse data, hopefully you were able to appreciate that NKT cells are rapidly activated. So within an hour and a half to 2 hours, you can detect cytokine in the serum after activating NKT cells. And they have potent effector functions. So they make cytokines as well as mediate cytotoxicity. And their subsets have distinct and overlapping abilities in which they mirror T helper subsets. So you have NKT1, NKT2, and NKT17, but their division is less rigid. And their differentiation is thought to occur during development. And you can have some plasticity in response, meaning NKT1s primarily produce interferon gamma and mediate cytotoxicity, but they can also produce IL-4. And their plasticity in response can be due to their tissue of residence. So NKT cells are primarily tissue resident cells. And location matters, as well as the type of, or the subset. So NKT1s, again, are primarily interferon gamma producers and mediate cytotoxicity, whereas NKT2s produce high levels of IL-4, and NKT17s produce IL-17 type cytokines. And so a little bit into development, because this will help you for the next part of the talk. So NKT cells are selected on CD1D expressing cells, and they have these slam-slam homotypic interactions. And while NKT cells were previously thought to proceed, their development was thought to proceed in a linear fashion. Now they're thought to originate from this precursor. Once you have committed NKT cells that you can delineate by using tetramers. So we know that this population of cells are committed NKT cells. And then we can divide them into their subsets based on classic markers. And so again, I mentioned the NKT1s, NKT2s, and NKT17s. And again, they can be defined by their transcription factor expression. So while the progenitor NKT cells, or these precursor population, can proceed into these various subsets, it doesn't have to proceed in a linear fashion where you go from stage 0 to stage 1 to 2 or 3. This progenitor population can either be NKT1s, 2s, or 17s. But based on this old nomenclature, that's how I'll describe the population. So we have our NKT1s that are stage 3, and then our NKT2s and 17s, which are stage 2. And so what I really want you to focus on for this part of the talk is that we want NKT1s, or these stage 3 NKT1s for cancer. And so we want to compare this subset to the other NKT cell subsets to see if their development, not their development, but their activation and responses are impacted by co-stimulatory molecules. Okay, so again, our overarching hypothesis for this section is that NKT cell subsets have different co-stimulatory requirements. And we wanted to look at the CTLA-4 CD28 co-stimulatory axis. So the prototypical co-stimulatory molecule is CD28. And so in our thymic mature, committed NKT cell population, what we really want to investigate is the role of CD28 and CTLA4 on NKT cell responses. Because CTLA4, as I'm sure you know, competes with CD28 following T cell activation as well as NKT cell activation. CTLA4 is quickly expressed on the surface, and again, it competes with binding to CD80-86. And what's been previously shown is that CTLA4 expression is low in NKT cells. And this is data from a science paper where alpha-gal-ser, the potent NKT cell agonist, was being described or defined. And what we found really intriguing was if you focus here on panel B and you look at antibodies against B7-1 and B7-2 and compare that to antibodies against CD28 and CTLA4, all three of these seem to inhibit splenic NKT cell function. So we wondered about immune checkpoints, right? Because that's key right now, using antibodies against CTLA4 and PD-1. And here, this would suggest that if you use an anti-CTLA4 or anti-CD28, it would have a similar impact on NKT cell function. And so again, we wanted to look at this further. Also, there's been really beautiful work looking at knockout mice, looking at what happens if you knock out CD28 or CD80-86, and it's been shown to reduce NKT cell numbers. But we were interested in activation, not in developmental cues, and that the role that these factors would have on NKT cell activation. So to do this, a student who recently graduated from the lab, Susanna, developed a proliferation assay. So first we take splenocytes and use those as feeders from genetic markers, load them with alpha-gal-ser in order to induce NKT cell activation. And then we take thymic committed NKT cells and enrich this population. We load with a proliferation dye and culture these thymic NKT cells with our alpha-gal-ser loaded splenocytes for 2 to 3 days, and then look at NKT cell proliferation. So that was the setup. This is the actual data. So again, we have our NKT cell subsets. After activation with alpha-gal-ser, you see nice, robust expansion. And we can look at this by gating on the population, and it's also shown here with the histogram. So we have a nice way to measure NKT cell activation and proliferation. So again, I mentioned that we want to look at the role of CD28 and CTLA4 in this. So to do this, we use a panel of antibodies, and all of our controls are shown here. So we have our vehicle control, right? These are negative controls. Then we have alpha-gal-ser, which would induce NKT cell activation. And then if we use anti-CTLA4, we would block CTLA4. So we would expect to get more division or more proliferation, right? Excuse me. If we add a secondary antibody, that would cross-link CTLA4, so we would get activation. So if we're activating CTLA4, we would expect to have less division, right? If we use CTLA4 Ig, that protein would bind to CD80-86, right? So then we would block signaling of both of these. And so since we're losing that activation signal, we would expect to see less activation, right? And then we could use a secondary antibody and cross-link, and again, we would expect to see less activation. So again, I'm just trying to show you that we had all of the controls in here, and I'll have the schematic throughout for clarity. So first of all, what happens if we activate our thymic NKT cells with alpha-gal-ser? Again, it works. We get nice expansion. If you look at the percentages, if you look at absolute numbers, as well as fold expansion. As you would imagine, alpha-gal-ser stimulates NKT cells. Right, so we have the system, it's working. So what if we activate with CTLA4? So let me see if sorry, I skipped the slide. So first we block. So we used anti-CTLA4 antibody. Again, if you use Alpha-Gal Serra, you get nice, robust expansion, activation. But if we block using an antibody against CTLA-4, there was no effect. So this was surprising. And you see this with looking at percentages, absolute number, as well as fold expansion. So in contrast to what we saw using splenocytes or what was published, it didn't seem to have any effect. And it's, sorry, my slides aren't progressing. And then they progress too much. Oh, okay. So then what if we had a secondary antibody and cross-link? So in this case, again, we'll use alpha-Gal Serra, we get nice expansion. But if we block, sorry, if we add in a secondary antibody and activate CTLA-4, we see no effect. So similar to what we saw when we use a blocking antibody or anti-CTLA-4, we see the same thing. So percentages, numbers, fold expansion, no change if we activate or block CTLA-4. So if you get negative data, keep working, right? That's the results that Susanna got initially. So now we wanted to look to see what happens if we block CD28. So again, use alpha-Gal Serra, nice expansion, anti-CTLA-4, no effect. But if we block CD28 signaling by using CTLA-4 Ig, we lose NKT cell responses. So while CTLA-4 doesn't seem to be playing a big role, if we look at the percentages or numbers or fold expansion, if we block CD28, we lose NKT cell activation. So they seem to be explicitly sensitive to CD28 in contrast to CTLA-4. Oh. Sorry, I don't know. Okay, why it's sticking, but it's okay. So now where do we go from here, right? I've shown you that blocking CTLA-4 and CD28. So in some, blocking CD28 signaling seemed to have a profound effect. So where do we go from here? So we can take this data and use what's classically used to examine T cell biology and do some math and be able to get the proliferation curve as well as the mean division number. So we know the cells that we collect in the end, right? So we start with a set number of cells. We can induce proliferation or block. Then we can look to see how many times NKT cells divided, right? We know the number of divisions. So based on this, we can again calculate the precursor proliferation curve. So how many times did that initial precursor divide, as well as see if there's a change in the average number of times the cells divide. So what do the data look like? When we block CD28 by using CTLA-4 Ig, and we look at the number of precursor NKT cells that are dividing, we see that CD28 blockade has a profound effect. So if you look at the slope of the curve, it looks like the vehicle control, right? Compared to stimulating with alpha-Gal Serra. And CTLA-4 has no effect here. But if we block CD28, again, we completely change the shape of the curve and we lose proliferation. And you can also see that with the mean division number. So that was for the bulk population. So then we wondered if there were specific effects on the different NKT cell subsets. So again, we were able to distinguish based on their transcription factor expression as well as their cell surface expression of NK1.1. So our stage 3, or remember our NKT1s that we want for cancer immunotherapy are called stage 3, and then the other subsets, the NKT2s and 17s, are stage 2. So now, when we activate with alpha-Gal Serra, again, we get robust expansion. But if you just focus on the red bar to the gray bar and look at stage 3 NKT cells versus stage 2, we see that stage 3, the ones that we want, divide less than stage 2. And you can look at the mean division number here And again, if you look at the proliferation curve, after activation, you get really nice expansion of stage 2, but not as much as stage 3. So what happens when you block CD28 signaling? So again, the raw data are at the top, looking at both the stage 3, that would be NK1.1 positive population, to the NK1.1 negative population. And if you look at the bars here, it's easier to see comparing the stage 2 to the stage 3. But if you look, when we block CD28 signaling by using CTLA-4 Ig, we lose proliferation. And it blocks both stage 2 and stage 3. So if we take a look more closely at the population to see what happens between the stage 2 and the stage 3 when we block CD28. And just for clarity, I have the arrows here. So again, look at stage 3, the NKTs that we want compared to stage 2. Both are impacted, but you see a significant or more significant reduction in the stage 3 NKT1 subset. And you can also look at the precursor curve to see how it's impacted. And you see in both cases, it's really, really flat, but we really lose that stage 3 population that we want. And again, blocking CD28 completely suppresses the curve, completely suppresses their proliferation. So we were excited about this, and we could look at the different subsets, and we found a impactful role for CD28 signaling. So we submitted the paper, and then the reviewers came back with really great questions. They said, first of all, when you activate T cells, you get downregulation of cell surface markers like NK1.1. So is the population you're looking at really stage 3 NKT cells? And then what happens if you use different doses of antigen? And so these were great questions, and we decided to look at this further. And so instead of enriching the population, we used tetramers and we sorted using FACS. So we sorted stage 2 and stage 3 NKT cells, loaded with the proliferation dye, and then we titrated the antigen. So before, we did all of the experiments with 10 nanograms per mil. So in these studies, we looked at 0.1, 110, and 100, and looked to see if NKT cells were proliferating, or if the antigen dose had an impact on their proliferation. So this schematic just shows you what we were expecting with our population. So again, we use flow to sort, and these are black 6 mice. So we had most of the cells were stage 3 and NK1.1 positive, and then we had our stage 2. And this may be the most beautiful negative data we've ever had. So as the reviewer suggested, when you activate NKT cells, there was a striking reduction in NK1.1, and you can see that clearly shown here with the MFI. But our data held true, which is great, right? So we pre-sorted the populations, the stage 2 and the stage 3 NKT cells by FACS, and then set up our experiments the same way that we did before. And what we found was there was still the significant reduction in stage 3 after blocking CD28 signaling. And you can see that with fold expansion, with the mean division score, and with the precursor frequency. Again, both stage 2 and stage 3 are suppressed, but what we found initially using the bulk population and staining with FACS later was still held up. So the data are the data, right? When the reviewers come back and ask for this, do the experiments. It'll teach you so much. And again, whatever you get is what you get, right? So that's what we found. And then when we titrated alpha-gal ser, interestingly, we found that the stage 3 NKT cells only start proliferating when you have 10 nanograms. However, stage 2, remember these stage 2 are producing both Th2 type cytokines and Th17 type cytokines. They start proliferating with just 1 nanogram. per mil of antigen. So our stage 2 NKT cells respond to lower antigen and expand more, right, than our stage 3. And so the mean division score is shown here, and then fold expansion. But again, you can see this very nicely when you look at the absolute number of live cells that the stage 3 stage 2 take off, whereas it takes a longer time for our stage 3 cells to start proliferating. So I showed you all of that data in vitro, so you may be wondering, does this hold true in vivo? So Susanna injected the mice with alpha-gal ser IP and then treated with CTLA-4 IG. Sorry, alpha-gal ser was injected IV, and then CTLA-4 IG was injected intraperitoneally. And we measured proliferation by looking at Ki-67 expression. And so the raw data are shown here at the top, where you see our stage 3 and stage 2 NKT cells. And what I think you can really see here is that what we saw in vitro held up in vivo. So when you block CD28 signaling, you get a striking reduction of NKT cell activation in vivo. So our stage 3 are significantly impacted here when you block CD28. And then graphically, we have the percentages shown below. But again, if you look here and compare it to here, you see a reduction. Okay, so I showed you all this proliferation data. You may say, okay, that's great, you can do that, but what about other functions? So we looked at cytokine production. And so as you would imagine, our stage 3 NKT cells are producing high levels of interferon gamma, much higher than the stage 2, and that's shown here. And you see an impact based on alpha-gal ser or antigen dose, right? And again, if you block CD28, you lose this. In contrast, the stage 2 make much higher levels of IL-4. But again, if you block CD28, you lose the cytokine production. The ratios are shown here. And then we also included TNF alpha. So not only do you see the striking reduction in interferon gamma, you also lose other cytokines such as TNF alpha. So the second summary is that we've observed baseline differences in NKT cell expansion or proliferation after activation. And we found that thymic NKT cells are refractory to CTLA-4 blockade or activation, but they are very, very sensitive to blocking or inhibiting CD28 signaling. And the population that we want to focus on, our NKT1s or stage 3 NKT cells, expand less and are more sensitive to CD28 blockade. In addition, they require more antigen to respond. So this tells us that we need to really tailor our therapies to make sure that we can harness the potential of these cells. Okay, so my last little part is to tell you how things may not go according to plan, but again, they all work out in the end. So this part is titled Individualized Genetic Polymorphisms Modeled in Different Mouse Strains Affect NKT Cell Development. So this was my graduate student plan. She thought, you keep working, things will work out. But in reality, you have all these dips and dives, and it's all about the journey, right? So this part is focused on breast cancer and BRCA1, and that's because it's estimated in 2022 that nearly 300,000 new cases of breast cancer will be diagnosed in the US. And 5 to 10% of breast cancer cases are caused by mutations in BRCA or breast cancer susceptibility genes. And so what this means is, you may have heard of this, 1 in 8 women are expected to develop breast cancer in their lifetime, right? That's 12%, 1 in 8. However, if you have mutations in BRCA1 or BRCA2, this significantly increases your risk for developing breast and ovarian cancer. And that's because BRCA functions as a tumor suppressor gene, and it's also involved in the DNA damage response. And many of these mutations that are observed in BRCA are in exons 11 through 13, which is unstructured. It contains 65% of the BRCA protein sequence. It contains the nuclear localization sequences, as well as it's involved in the DNA damage response. So we wanted to use BRCA mutant mice as a model to study breast cancer and the role of NKT cells in mice that have BRCA mutations. And this was bolstered by this finding. So in our clinic, we were able to obtain patients' samples from patients that had mutations in BRCA but didn't have cancer, and compare that to patients with breast cancer. And so we were looking at NKT cells in the blood of healthy donors. These are female healthy donors, as well as breast cancer patients and patients with mutations in BRCA. And what we found was that when we looked in the blood of patients that had BRCA mutations, we were unable to detect NKT cells in their blood. So this highlighted a potential role for BRCA in regulating or being important for NKT cell responses in BRCA-associated breast cancer. So we had BRCA mice that had a FLOX allele. They had mutations in exon 11, and we looked in the thymus, spleen, and liver, as well as the lymph nodes and bone marrow. But here I'm showing you thymus, spleen, and liver. And if you compare to C57 Black 6 mice, mice with mutations in BRCA had a profound reduction in NKT cells across the board. So if you look by percentages as well as absolute number. And so then we looked to see if there was a particular stage of development in which NKT cells were stalled or stuck. And compared to C57 Black 6 mice, in which, as I stated before, most of the NKTs are in stage 3. If you look in BRCA mice, they mostly appear to be stage 2 NKT cells. And again, by percentages as well as absolute number. So Susanna was excited. We were working on this. We performed like bone marrow chimera experiments. She used a retroviral Cree in order to look at NKT cell development to see if BRCA was playing a role. And then we took this TEL SNPs and sent them to DART mouse and found that even though we were told that the mice had been backcrossed for more than 10 generations, they really weren't. So look at this yellow portion, right? This was non-Black 6-like, which is really high. And so we were using this mice, but they weren't on a Black 6 background. And so we also had the SNP information. So Suzanne investigated how the mice were derived, re-derived the strain, and then looked to see if there were differences. And so if we looked at the mice that just have the FLOX allele compared to those with the Delta 11, so with the mutation in exon 11, now you see there's no change. So can you believe this? It took her 2.5 years to re-derive the strain, and we completely lost the phenotype. Even if we look at subsets, there was no difference. So this slide was taken from her defense. Where she wondered what happened, where did the phenotype go, right? And so she did her due diligence, looked to see how the strains were originally derived from the MMTV Cree, and found that FVBN mice are often used because their nuclei are huge. And so it's much easier to make transgenic animals using this strain. And so when we compared C57 Black 6 mice to FVBN mice and our BRCA floxed, and then the ones with the mutations in BRCA, we found that the strain of the mouse, as been shown with Black 6 and BALB/c, really impacts the NKT cell population. So again, if you just compare Black 6 to FVBN, you'll see that FVBN mice, have a really high percentage of NKT cells, right, in the thymus compared to the other strains of mice. And you can look at the different subsets, right? And then our NKT1s, 2s, and 17s, and you look at the different subsets. So then we wondered, what are the potential mechanisms? So we know that the T cell receptor can have an impact. And so we looked at V beta 8, and it's known that FVBN mice are V beta 8 negative, right? And NKT cells, or iNKT cells type 1, the ones that we study, are V beta 8, 7, and 2 positive, right? So they already have a different repertoire of NKT cells. And interestingly, we did find that they did have V beta 7 and V beta 2 NKT cells, but there's this negative population here, right? That's not 8, 7, or 2, that is tetramer positive. And so this could be a factor impacting them. We also looked at CD1D and found that the FVBN mice express lower levels. And if you look at the MFI, you can see that here. So FVBN mice have a different TCR repertoire. They have a decrease in CD1D. And then we wondered about TCR signaling, because strong TCR signaling has been shown to promote NKT1s and NKT17 development. And so we looked at EGR2. And again, if you compare C57 Black 6 to FVBN, you see it's much higher in each of the subsets of NKT cells. So we've identified multiple factors that could be playing a role in this difference in the NKT cell subsets that we observed in the mice. So this is the last part. Again, I just want to show you how you could really make sure that you know what you're working with, right? So we did all this work in the mice, but we ended up finding or discovering potential mechanisms that could tell us why there was a difference in the NKT cell population in our animals. So first of all, we found that the BRCA1 mutation did not impact NKT cell development when we used appropriate littermate controls. We did find that genetic polymorphisms can influence the NKT cell population, and compared to Black 6 mice, FVBN have an increase in type 1 NKT cells, and they have a significantly different subset distribution of NKT cells. So overall, I told you 3 different stories about NKT cells, and we feel that they're not being exploited to their full potential in cancer immunotherapy. We know that genetic factors can influence this population, and that the different subsets have distinct requirements for their activation, and that when we activate with alpha-gal SER, we know that TCR stimulation with a high affinity ligand does enhance NKT1 activation. So we need to do more research to really understand how to best utilize this unique population of cells. So I know I went on and on about NKT cells, and if you didn't get it all, it's okay. So I have this quote from Maya Angelou saying that I've learned that people will forget what you said. So hopefully you won't forget it, but if you do, it's okay. And then people will forget what you did, but they will never forget how you made them feel, right? So when you're in the lab, remember your purpose. And of course, I couldn't do all of this amazing work without my outstanding trainees. I would like to thank all the patients that donated their samples, all of the interns in my lab, my wonderful collaborators, and of course, you need support. So I'd be happy to take any questions you may have. Thank you.
Hi, Tonya.
Hi.
You can start. So, great talk and very interesting story. So, I'm going to start with a question from the first story about the GD3. So, is this gangliocyte unique for the ovarian cancer? Something about the peritoneum, or can you see it in different type of cancer, like a lung cancer or something? And also, have you tried to have and make antibodies, blocking antibodies for these gangliocytes, and as a way to increase the NKT in cancer treatment?
Those are great questions. So we do think that this GD3 is different. From the TLC, we saw 2 bands. And so we think that it's modified, and we tried to see if it was acetylated. We need to do more mass spec in order to really pin down, because other people have found an activating form of GD3. And so if you're a lipid biochemist, you're saying GD3 is GD3, right? Everyone's a GD, but we've isolated it from the brain, from bovine milk, like buttermilk, as well as from the patient samples. And we found different functional difference. We found differences in function, even using the same concentration. So we think that this one is modified, but it's found in breast cancers as well as melanoma and renal cell carcinoma. And we've used antibodies. We've treated our ascites with antibodies, and we lose the inhibitory effect. So we do think that antibodies could work, but not as a single agent. So the same antibody that we use, that clone has been used in clinical trials, and it has not worked well. So we think that we need to do, that's why we started looking at VEGF. We think there needs to be some combination in order for it to be effective.
So you need to activate, in addition to eliminate the repressing signal, you have to do. Okay. All right. Yes. Thank you.
Thank you.
Dr. Webb, question.
Yes.
So first of all, thank you for a great story. You've always been a great storyteller. So what I'm going to remember is how you made me feel going through this great story. So thank you. So a couple of questions regarding NKTs, right, and their potential use as a cancer immunotherapy. Is it known whether these NKTs have specific homing properties, such as, resident memory T cells that tend to kind of reside and go back to their place of origin? So do NKTs behave similarly?
So we were looking at mature thymic NKT cells, thinking that those cells still have the potential to go to tissue, and they do. So they express chemokine receptors, and they're thought to be mostly tissue resident. So the ones in the liver are the ones that mediate anti-tumor activity, at least in mice. So it's been shown that liver, if you take, there was a JXMed paper by Crow et al. That showed if you isolate NKT cells from the thymus, liver, and spleen, inject those into mice, the ones in the liver are the ones that cause tumor regression.
I see. So if it's back in the liver, though.
Yes.
Okay. And then the second question I have is regarding whether these NKT cells are susceptible to the same mechanisms of exhaustion as conventional T cells. I know you showed CTLA4 and CD28 data, but do these T cells exhaust through PD1, TIM3, LAG3, etc.?
Yes. So you can block with antibodies against PD1 in order to restore NKT cell function. So yes, they do utilize the same mechanisms. And we've looked in breast cancer patients to see the differences between patients with functional NKT cells versus those with non-functional NKT cells. And those with non-functional NKT cells had an upregulation of those exhaustion markers.
Okay. And if no one else has a question, I have one more. So in the CD4 negative NKTs, that means that they can't recruit in LCK to the TCR in immunological synapse, and therefore those downstream enzymes and phosphatases and kinases are not activated, right? So go ahead.
Go ahead. I'll let you
My next question was, and related to that is, what is the role of the individual CD3 subunits in these CD4 negative NKTs?
Oh, so I haven't thought about it in that context. So I shook my head too fast. Oh, that's a great question. I need to think about it because in my mind, NKT cells signal, I'm sure that there's a straightforward explanation for this, but it is not coming to me right now. I'm like Laurent, or I'm going to call on other people. I can't believe it, but I'll think about it and I'll get back to you. You're saying how does CD3 signal within NKT cells?
Yeah, just wondering if there is a known role for each of the three CD3 subunits. That's part one. And part two is in these CD4 negatives, you know, CD4 is responsible for bringing in LCK into the synapse. And if you lack CD4, you know, I'm just wondering what is initiating the TCR signal in those cells. But something we can think about.
Yes, I just haven't thought about it that way. That's a great question. And I'm sure as soon as I get off, I'm like, oh my goodness. But I'll follow up with you directly.
Thank you.
Yes,
Thank
I'll
you.
think about it. But thank you I haven't thought about it that way before. Thank you for asking me something I have not heard and don't have a direct answer. Okay.
I have a question.
Yes.
So you showed that if you dependent on the TCR V beta repertoire in the NKTs, that you'll have whatever subsets. So is that a developmental thing, or if I took out NKTs and were able to engineer them with whichever TCR V beta chain makes like stage 1, would it then become a stage 1 NKT?
Oh, thanks. I'm thinking about your question as well as Eduardo's question. I'm still thinking about that because actually we've looked like at ZAP70 and SLIP70, you still get the same signaling in both populations. And back to your question, there's really nice work from a group at UCLA, Lillian, where she took iPSCs, right? Progenitor cells, and then put in the NKT cell T cell receptor, and you're able to get signaling. So she can take cells that are stem-like cells and use those for iNKT CAR therapy by putting in the T cell receptor. So you're asking about the individual chains, and we've done that. You can do that in Jurkat cells. You can put in the NKT cell T cell receptor and get it to signal. So you still have the downstream signaling events. And I need to look at I don't think we looked at the individual domains of CD3. But you do still get the downstream signaling, Eduardo. I need to see what induces the signaling, but yes, you still get all the downstream signaling from the T cell receptor.
Great, thank you. And Joy, was your question answered? I know we're kind of 3 minutes past the hour, and I don't know if you have other meetings, Tonya.
Oh, it's okay. I mean, I do have in a little bit, but Joey, did I answer your question?
I was just wondering more of that the subtype differentiate. Well, so real quick before everyone leaves, there's a survey that was posted in the chat by Dr. Cota Gomez, if everybody could fill that out. And then also my question was just, so the subtype differentiation based off the TCR V beta chain that's in the NKT. Mm-hmm. So is that a developmental thing, or can that be changed if you introduce that TCR type into the NKTs later?
Oh, so you're saying, so it's the differences between NKT 1s, 2s, and 17s is independent of V beta 8, 7, or 2. That is their affinity for alpha gal stere, but they can be 8, 7, or 2 and still make interferon gamma, IL-4, and IL-17.
Okay, thank you.
All right, well, Dr. Webb, thank you again for attending. We very much enjoyed your talk. So I hope you enjoy the rest of your talks with the folks you're meeting today. And then maybe we can set up a time to catch up later, either AAI or AACR.
Okay, that would be great. Thanks again. Thanks so much.
Bye-bye, Tonya. It was very nice seeing you. Bye-bye.
You too. Thank you. Bye.